Welcome to today's webinar on ethealthworld.com. Today, today we have with us Dr. Vineet Datta, who is responsible for executing appropriate healthcare strategies and clinical operations in preventive healthcare for the Apollo Group of Hospitals. He is also the member of the College of Emergency Medicine, London. Dr. Datta is an elected Fellow of the Royal College of Physicians and Surgeons, Glasgow, as well as Fellow of the Royal Society for Public Health.
Open the PPT. And you do it open the PPT. The potential for personalized medicine to transform clinical practice has been a subject of intense debate over the last few years. We understand that no two individuals have the same genomic material, yet every person has a unique variation of their own genome. Yet most of the variations that we find in individuals has no effect on health. An individual's health stems really from the genetic variation with behaviors and influences from the environment. Personalized medicine is really a medical model that proposes the customization of healthcare, really where the decision-making, medical practice, the solutions we provide to our patients are really tailor-made for every individual. In this structure, the diagnostic testing is often employed to select optimal therapies in context of the patient's genetic content or his cellular behavior. One way in which the biological variation makes itself quite clear is how one responds to medications or drugs. And intends to use predictive tools to evaluate health risks and design personalized healthcare solutions that help mitigate patient risks, prevent disease, and even treat with precision when it's required. So good afternoon, colleagues. Over the next 30 to 45 minutes, I'm going to be sharing my thoughts on the personal genome, what's the future of healthcare in India. As a wise man once said, your genetics load the gun, yet it's your lifestyle that pulls the trigger. So over the course of the next few minutes, we'll work our way through what's the current burden of disease within the country, especially from the non-communicable disease standpoint. Where do we stand on personalized healthcare and genomics? What's the value in the interpretation that we can take out in terms of the medications that we prescribe to our patients or the cancers those patients fight and we help them fight them? Every day, millions of people take medications, some of which will not help them. It's important that we recognize that physicians need to take individual variability into account when we're dealing with precision medicine. And largely, personalized medicine has moved across from traditionally having doctors use family history, socioeconomic circumstances, environmental factors, to now really looking at the genomic and the genetic testing. So what's the application in it for our patients, and where do we stand on that debate? So just to give you an overview in terms of, and I'm sure you know this, is really where do we stand on the dual burden of disease, especially for our country. Whereas for us, the infectious diseases have certainly not gone away, we are labored with fighting the menace of cardiovascular disease, diabetes, cancer, obesity, and mental health. Globally, they account for over 38 million deaths every year, and a vast majority of them are lifestyle related and preventable if detected early during the insidious stage of development, or may even be controllable if not curable in some stages, in some instances. The burden of disease of non-communicable infestations accounts for over 3/4 of the total deaths. And India is very much accountable for over 17% of those deaths. Cardiovascular disease accounts for almost 1/4 of the deaths that take place within the country. We have over 500,000 people who die because of cancer every year. And in diabetic patients, 2/3 die from cardiovascular disease and stroke. Interestingly, the onset of cardiovascular disease is about a decade earlier than what we see, for example, in the United States. Cancer is the new menace that we taking a grip of. Don't make it with late detection. Often half of the cancers that are associated with us involve lifestyle choices we make, whether it's obesity or tobacco use. And we know for a fact that the success rate in cancer treatment is almost in excess of 2/3 of 70% if the disease is diagnosed in the early stages, and that significantly comes down if it's intervened in the later stages. In fact, 1 woman dies out of breast cancer every 10 minutes in the country. That's shocking and both appalling. By the time we're through with our webinar today, 6 women somewhere within the country would have died because of breast cancer. In fact, Globacon, which is an initiative on behalf of the World Health Organization's International Agency for Research on Cancer, suggests that we may have 1 death every 6 minutes in the coming 10 years from cervical cancer. The situation isn't really very different whether we look at obesity, where we're the 3rd largest obese population in the world, or in the case of mental health, where clearly there's a significant burden of mental disorders, especially in the young adults in the 15 to 45 population, which is the most socioeconomically productive population or section of our community. So what do we really mean by personalization of care, and what does it really entail for us? So personalized healthcare really puts the patient at the center of the entire healthcare experience for us. And that's what significantly impacts both the delivery, the cost model, as well as the quality of healthcare being provided. So the patient is the center of all activities that are provided, whether it's through a physician, a nursing support, through associate physician support in terms of specialists, whether we're looking at lifestyle choices or indeed rehabilitation through physios. Personalized medicine, in comparison to healthcare, really refers to more specifically the use of genomics and genetics. So how can we look at, for example, predisposition of an individual to a disease? What can we make out of an individual genome that an individual harbors? What are the variations of behavior that we can see between genes and the influences of environment or population subsets? How is it that an individual reacts differently to medications, something we'll discuss under pharmacogenomics during the course of the day today. And then really, what's really caught the eye has been the availability of direct-to-consumer genetic tests which look at predisposition of diseases. So the applications of personalized medicine really look at diagnosis, risks and intervention. They look at drug development and usage, and also take into account cancer genomics. So in essence, to summarize, the personalization of healthcare is really providing the right treatment to the right patient at the right time. The basis of all this is really what we call as GWAS, or the Genome-Wide Association Study, and I'll take you through the 2 sets of images that we have. But really, in order for our physicians to know whether a mutation is connected to a certain disease, researchers often undertake what is known as a GWAS. So this study will look at really one disease, sequence out the genome of many patients with that particular disease to really look for any shared mutations in the genome. The mutations that are then determined to be related to the disease by this study can be used to diagnose the disease for future patients by looking at genes by genome sequencing to find the similar mutation. The first GWAS study was conducted over 10 years ago in 2005, when it looked at age-related macular degeneration, and as of last year, well over 1,300 GWAS studies have been completed. Although the cost of whole genome sequencing is dropping significantly, it still is too expensive to consider sequencing of a large number of genomes required to observe a trend of disease association with a particular location in that genome.
So really what GWAS focuses on is an SNP, or a single nucleotide polymorphism, a single nucleotide. And by studying these, they're able to analyze a few 100,000 nucleotides rather than 3 billion odd that they would need to, that compose the whole genome. And if you look at the image on the left, the basic design of the GWAS is very simple. It looks at dividing participants into really two groups: patients with a disease or a trait of interest, or patients without that disease, which we may call the control group. The SNPs then are really analyzed and compared across both groups. And what the researchers hope to identify is really the presence of that particular SNP present in the disease group that's not present in the control group. So if such an SNP or a polymorphism is located, then researchers have identified a genetic sequence that's in association with a particular disease. And by collecting increasing amount of genetic data and DNA sequence data, researchers can then look at analyzing trends if those particular alleles are indeed correlated with a particular disease. So as I was mentioning, thousands of disease and traits have been investigated, and things range from type 2 diabetes to epilepsy, looking at Alzheimer's, Crohn's disease, breast cancer, asthma. And GWAS, as I mentioned, are really the basis of most direct-to-consumer genetic companies and the solutions that they provide to the customers that they serve. Without drawing too many conclusions out of graph, I think it's also important to understand that the relationship of identified SNPs to a particular disease is only an association. Certainly, some people will have the risk variant and not have the disease, while others will have the disease while still not have the variant in place. As a result, GWAS often presents itself in terms of an odds ratio, in terms of the measure of that event happening in that one group compared to whether the same event is going to happen in the other group or not. There are a number of reasons why the GWAS produce these odds in condition, because we need to understand that most traits or diseases are impacted by environment, the lifestyle we lead, not just genetics. And into that process, it may not be one gene, but multi-gene involvement which could have that structure. What would also be important is that the odds encompass the risk of getting a disease over the entire lifestyle or over the lifetime, even though the risk may change during an individual's lifetime. So you have a much lower risk of having, for example, colon cancer when you're 55 and 60, and a much higher risk, almost extent to 2/3, if you're towards the end of your life, and that's in terms of an age group structure. Again, so what we need to understand is GWAS is really a statistical tool. It's used for research, increasingly used for clinical application. It is looking at SNPs, or single nucleotide polymorphisms, into the structure. We do need to, however, understand, and it's important to acknowledge, the fact that we calculate the probability of developing a disease when we look at such tests compared to the average population. And this probability is usually backed by a statistical foundation or scientific research in that process. So what have been the adoption of genomics as we look across the structure? And many of you may have heard of the famous story of 23andMe, a personal genomics and biotechnology company that was founded in California in 2006. Certainly was looking at servicing health, ancestry, and complete edition genomics to their patients. And in fact, in 2010, the FDA in the US acknowledged the use of genomics and personal genome testing, or genome services, as a device which needed federal approval. So in 2013, the US FDA in fact ordered 23andMe to discontinue and market their personal genomic service solutions. And one of the reasons for that was the direct-to-consumer approach, without having intermediary of healthcare professionals like physicians to explain the difference between the diagnosis and the association, the repercussions and the counseling aspects of what would be the impact of the tests that were coming through in that process. That being said, in 2014, the personal genomic service by 23andMe is available in Canada. It's available in the United Kingdom. It's received its CE certificate, CE mark from in the UK, and it's also been accredited by the UK Medical Healthcare Products and Regulatory Agency. In fact, I think just last month it's enrolled its 1 millionth genotype customer in that process. I think what we do need to understand as we look at aspects of genomics, which are really health related, the aim and the vision is to see, is there a better way to better manage health and lifestyle and understand what some of those aspects are. Certainly within the country, we have a number of companies like Datar and Sapien, which look at providing a genetic analysis, molecular pathology, health risk screening across the spectrum of various solutions that are available within India. So why is really there so much of a fuss when we come to the personal genomic analysis? I think it's important to understand that specific genes have been implicated in diseases or disorders, and can they help us to better judge the manner in which the cellular mechanism is working. The analysis can help in hopefully giving us timely warning of potential threats to health. An individual's risk of getting a particular condition or disease is given as a comparison to risk in a general population. So the word risk really implies a chance or a probability of developing that condition. Yet it's important to understand that risk doesn't mean the presence of disease and certainty of developing the disease, because both genetic as well as environmental factors, as I mentioned earlier, are responsible in their onset. So personal genomic analysis within the structure of that process is looking at a number of different conditions that can be analyzed either through a person's blood or the saliva or the tissue, even something like hair. So diseases like Alzheimer's, asthma, diabetes, hypertension, myocardial infarction, cancers like breast cancer, lung cancer, prostate cancer, how the body reacts to metabolism in terms of whether we look at alcohol or vitamins, what about genetic conditions in terms of how one responds, for example, to addiction, whether it's nicotine, other disorders like sickle cell disease or thalassemia often are integral to that process. So really the research in terms of what we're looking at is really to try and define risk. Is there a way we can look at enhanced classification, and can we come up with innovative therapy in that process? And really when we look at that process, it's really intended to have a better traction and an outcome between a physician and his patient in terms of that relationship, which is why the direct consumer has always run into those challenges. So if you have a physician or a doctor as part of that process, the report or the analysis that one gets can prove to be an invaluable tool that can give the doctor insight into the patient's genetic mechanism, and then can hopefully help the doctor conduct tests or examinations that are well directed or analyzed to have better clarity in that process. So as I was mentioning, it's really looking at more in terms of identifying major components of disease involvement. Is it possible to catch those and detect those in an early phase? And then, if that's possible, can you institute timely treatment and optimize lifestyle or surveillance measures in place to make that happen? Really, I think the advances in personalized medicine create more of a unified treatment approach specific to the individual and their genome. And personalized medicine provides better opportunities with that earlier intervention and even more efficient therapies and opportunities for treatment in that regard. Again, having the ability to look at a patient on an individual basis should hopefully allow a more accurate and a specific treatment plan to take place. Certainly genotyping is looking at one of those measures by which we take the biological assay. And a large number of companies like 23andMe and Navigenex and Illumina and others have actually taken that on board in that structure. But when we look at really from a value of a patient perspective or a potential customer who's going to undertake these tests, it's really looking at driving awareness of particular risk and the precautions that one can take. It's sort of driving home increased surveillance with the aim to reduce the risk of development of that particular condition. It's looking at assessment of genetic influence on metabolism, and then a physician insight into genetic mechanisms in place. So one of the main aspects where there's real value of how we can use genomics in that process is really identifying responders and non-responders to medications. Is there a way we can smartly use genomic analysis to avoid adverse events and even optimize drug therapy? And into that process, a large number of associations can be described, whether it's drug exposure or risk to adverse events, or whether it's mechanism of drug action alteration, or even a polymorphic drug target disposition in that process. So pharmacogenomics really, it's the study of genetic variation that gives rise to different responses to medications and drugs. And it's estimated that genetics can account from anywhere from 20 to 95% of variation in drug disposition and effects. Obviously, there are non-genetic factors that come into play. It's an individual in terms of his demographics, age, organ function, any concomitant therapy, drug interactions, the nature of disease, all of that certainly has an impact in place. An aspect of this is really looking at pharmacogenomics, where individual genomes can help us provide a more informed and tailor-made drug prescription. Often drugs are prescribed with the idea that they will work relatively the same for everyone. And in that application, there are a large number of factors that certainly need to be considered to ensure that one can prevent adverse events, allow for appropriate dosages, and also create a maximum efficiency of the medications that we are prescribing in that structure. In addition to specific treatment, personalized medicine can greatly aid in advancements of preventive care. So for instance, many women have already been genotyped for mutations like the BRCA1 and 2 gene. They're predisposed because of family history of breast cancer or ovarian cancer, for example, can be taken into account. As more causes of diseases are mapped out according to mutations that exist within a genome, the easier it can be to identify them within an individual, and measures can be taken to prevent those in place. Now, having an individual's genomic information can also have a significant impact in terms of developing of disease as they're waiting for approvals to take place, as one example may be. So having a detailed account of an individual genetic makeup could be a major asset in deciding if an individual should be chosen, included or excluded in final stages of a clinical trial, for example. Really what we're looking at as we look at pharmacogenomics is really moving away from the reactive approach in clinical care from the diagnosis to selecting a drug, waiting for the drug not to work, as the disease is progressing, switching the medication, and then taking account. Here we're looking at translating that down from a proactive process of looking at predisposition, looking at the diagnosis and the prognosis, and a right drug selection in that process, and then look at monitoring into that account. Certainly, I think with personalization of medicines, we can look at a way in which the often trial and error strategy can be limited. Treatments can be more specifically tailored to an individual. And personal genotype can allow physicians to have more detailed information that can guide them in terms of their decision making as part of that process. So it's really, again, looking at therapy with the right drug at the right dose in the right patient. One of the common examples of that, obviously, is you'd be aware of, is tamoxifen, commonly prescribed to women with ER-positive breast cancer. But 65% of women, as we know, initially taking those medications develop resistance. And research told us that certain mutation with the CYP2D6 gene, that was encoding the metabolizing enzyme, were not able to efficiently break down tamoxifen, making it an effective treatment for the cancers. Since then, women are now genotyped for their specific mutation, so that immediately these women can have the most effective treatment in place. What you see on the right, on the top, is really more in terms of other examples of how the effectiveness of drugs take shape. In fact, of the data that I have from the US again, almost 100,000 people die annually, and over 2 million are hospitalized because of adverse reactions and medications that take place. And really, this is an opportunity where we look at safer drug options, more exact dosing, optimum dosages in place in that structure. Certainly, in terms of almost about 6 to 7% of patients in hospitals experience a serious adverse drug event in that process. So how do we look at using some of that information in terms of the next aspects of pharmacogenomics to impact care for our patients? So one of the current common uses is really in patients who are infected with HIV. before prescribing the antiviral abrogate drugs, doctors will now routinely test HIV patients for genomic variants that make them more likely to have a bad reaction to the drug. Another example is Herceptin in breast cancer. This therapy works only for women whose tumors have a particular genetic profile that leads to overproduction of the protein HER2. Again, there are other examples in place as well, whether we're treating acute lymphoblastic leukemia and looking at mercaptopurines, or even other aspects like colon cancer and using irinotecan. One of the other interesting areas that's come about in the last few years is also to see how can we look at optimize and use pharmacogenomics for better outcomes. And certainly when we look at aspects of, for example, antiplatelet therapy. And clearly there are now solutions available that help us look at analyzing components of tailor-making our antiplatelet therapy, whether it's the genotype component or the platelet function component in that structure. We are aware that in the Indian population, studies show that almost up to 6 out of 10 Indians may carry an alteration on a functional variant of the particular gene in this regard, which makes a poor metabolizer of the CYP2C19 enzyme in that process. So it really takes into account whether there's an override or an underride of the drug in place, which can either lead to bleeding as a side effect or an underutilization of the antiplatelet function for patients who are undergoing an acute coronary syndrome or undergoing a PCI in that regard. And that's certainly important to bear in mind in that regard. I think certainly BRCA1, BRCA2, the human genes that are known to produce tumor suppressor proteins, again, specific inherited mutations increase the risk for female breast and ovarian cancer. They've been associated with risk of other types of cancers as well. And together, both account for 20 to 25% of hereditary cancers, breast cancer, and almost 10% of all breast cancers in that regard. So we mentioned a little bit in terms of how we were looking at the use or optimization of medications. We doing with pharmacogenomics, and there was a bit of an overlap as we looked at some other aspects of how this could be applied in terms of personalizing care for cancer research and treatment. And this really takes us to oncogenomics, where how can we look at a higher throughput of sequencing methods for characterized genes associated with cancer to better understand the disease pathology and to hopefully improve drug development. Again, Herceptin, classical example here, a monoclonal antibody drug that interferes with the HER2 receptor to have been used in treat certain kinds of breast cancer. Other examples like tyrosine kinase inhibitors like imatinib, which has been used again in CML or chronic myeloid leukemia. And all of these really help us to look at a rational drug designing, which can take place to personalize the level of care that we're providing in that structure. And certainly, I think oncogenomics is one of the most promising branches that looks at providing us greater insight and knowledge into the implications of drug therapy for the patients that we treat, whether it's through therapeutics or prognostic measures in place. What can be the other applications of personalized medicine in that process? Certainly aging. Chronological age is very different from the biological age. So where chronological age really represents the old as our years, biological aging refers to the aging of the body. And the condition is really linked to chromosomes after each cellular division. Biological aging is related to the length of telomeres. So it's really the stretches of the DNA at the end of our chromosomes. And when we're born with telomeres of a certain length, these get shorter as our cells divide, resulting in aging. And that's what scientists think. But we know that not all telomeres are born equal. Telomere lengths vary from person to person, generally similar in siblings, which may suggest that there is some degree of a genetic control in that process. But I think more importantly, what researchers have found is that individuals who carry specific variants to the smaller telomeres, that's less padding to the fragile genes, that's really where is the point of interest. So for example, in terms of the telomerase RNA component of the TERT gene, where earlier studies suggest that there may be a low risk expression association with a faster biological aging. Certainly other aspects that are interesting to note are what's the impact in terms of on our learning from our mistakes that we make. So we're often angry at ourselves when we're unable to learn from certain experiences. Numerous times we've made kind of the wrong decision. Its consequences can be unfavorable, but the cause really doesn't lie just in our thinking. It may be, well, a mutation, the DRD2 gene may well be responsible in that account. And this is the one which causes a smaller number of dopamine receptors. So they're responsible for remembering our wrong choices and then in turn enable us to make better decisions when we encounter a similar situation. Other applications, again, could look at things like, for example, inherited conditions like thalassemia. Certainly, there's options available for potential customers to look at genetic screening for couples, and you can look at thalassemia, hemophilia, muscular dystrophy, retinitis pigmentosa, and other renal disorders under that purview. So this really brings us to how we channelize some of these direct-to-consumer genomics and some of those tests that come into place. Certainly, I think there's value in how we look at drug delivery and drug delivery models, as well as the potential positive impact they can be on cancers. But it all focuses down on in terms of how this can be integrated with interface of a physician to guide a patient through that process. And again, we know for a fact that a focus on primary care, for example, has led to a 50% decline in cardiovascular mortality and almost a third reduction in terms of diabetic mortality over 24 months in the United Kingdom through that process. We're also aware that there are variations that exist in terms of health checks across health checks providers. And that's why, because no two individuals are the same and because we're all unique in that process, we understand that no two persons can ever have the same health status, the same demographics in terms of medical history, family history, social history, physical or mental well-being, as well as impact of genetics and lifestyle. And this is really where a personalization of a health check comes into place, which hopefully aims at providing a comprehensive screening program which is tailor-made for an individual. So the underlying demographics that I may have as an individual will vary from what you have. And so hence, why should our health checks really be the same? And that's really the envisioning of the personalized health check, which looks at having a physician up front understand variabilities of an individual patient or a customer, and then really customize investigations that can be made in that process. So investigations are really tailor-made in that account. Why should you have your blood group checked every year on a health check? That's not going to change. What is important to note is how can you look at standardizing the investigations that we offer to the patients or the customers that we serve. Again, largely it's got to be evidence-based care. So when do we start, for example, with the pap smear? So for asymptomatic individuals, are we starting at the age of 21, continuing to 65, for example, with a 3-year screening? And knowing fully well, we may be looking at an annual one if there's a CIN positive case. Mammography, for example, are we looking at an annual every 2 years over the age of 40 for somebody who's asymptomatic? Take into account if there is symptoms, family history, how's that being altered? There's been a large amount of debate in terms of overuse of tests, for example. So PSA again is a classical example. What's the value in a younger age group if we're taking that into account? Should we be looking at more of a 55 to 70 plus sort of family history population to make that into account? And the aim of personalizing all of this is looking at providing increased traction, again, of customization to an individual's need, so you can then develop a better and a more effective treatment plan, which is complemented by lifestyle changes in that process. I think when we're discussing genomics, I think our discussion wouldn't really be complete if we didn't take into account the whole debate of the direct-to-consumer tests that have been part of that system. And as personalized medicine is practiced more widely, a number of issues of discussion will certainly arise, and they will certainly look at issues like intellectual property rights, patient privacy and confidentiality, as well as regulatory oversight in that process. Again, the analysis of the diagnostic data in that personalized medicine space with its adoption will be an area of interest. It wasn't, in fact, too far ago as 2008 when Time magazine came out with the retail DNA test, which was started by 23andMe in that account, and certainly a lot has changed since then in that process. A number of proponents of consumer genomics, including direct-to-consumer genetic testing, have been made available. And when they started off, they were without the need for an intermediary medical professional. So direct access as an individual's right, noting potential benefits. However, increasingly more and more, we've seen the use of an overriding physician involvement in that process to help guide patients through that process, as well as providing appropriate counselor support, is really what's strengthened that whole process. Clearly, the report which came through from the FDA a couple of years ago, which was looking at paving the way for personalized medicine and the role of medical product development, certainly has an impact in terms of trying to integrate genetic biomarkers and information for clinical use and drug development. And certainly the institutionalization of a genomic reference library is an area of interest. I think the most critical issue as we look at commercialization of personalized medicine into that space is really protection of our patients. And into that space, it's really how do we address upfront issues with regards to psychological effects on patients due to the testing and the aftermaths of those results that come on board. Earlier this year, the FDA, in fact, has permitted marketing for the first direct-to-consumer genetic carrier test in the US for Bloom syndrome. So that's already in place, as well as there have been various statements that have come through, for example, from the American College of Medical Genetics and Genomics in that regard. I think for us it's important that we understand, we recognize the ethical guidelines in place to ensure that there is clear value of having a physician and a counselor in that process. So genetic counseling, the process of making sure that people who undertake such genomic analysis clearly do understand and have the ability to adapt to the medical, psychological, and the familial implications of what they're getting into. And clearly, that there's a pre and a post-test counseling that takes place, and there's an overriding supervision that takes place from a physician in that regard, which can create that value for the potential patient. So as we look ourselves as a country, one of the world's youngest superpowers, clearly and demographically, we have a large percentage of Indians that are younger than others in the rest part of the world. I think something that's been communicated quite avidly across the world. But I think it's also important that we ensure that we provide enough in terms of activity to override and move away from the non-communicable diseases that present a credible threat to our country in that regard. I think the silver lining in that process is that the human body is inherently forgiving. It sometimes does give second chances, unfortunately not to all. And into that process, it's important that we drive both ownership as well as information, as well as clarity to the patients in terms of the kind of care that we're providing to our patients. So I think genetic testing really in that process and personal genome analysis, it's an important medical tool in certain situations, certainly, and creates interest in healthy individuals in terms of looking at association with common complex diseases, and its potential in terms of integration with physician oversight, as I mentioned, which is clearly of a value. We, however, do need to understand that conditions and diseases are complex, almost all of them. They are multifactorial, and there will be impacts not just of genetics, but of biological, social, and environmental causes in that process. And while there's been intense debate and controversy that's come about through the direct-to-consumer genetic testing that's come on board, I think that there are clear benefits that can be obtained, providing we have enough safeguards in place to ensure that there is value that we're creating in terms of both information as well as safeguarding the rights of our patients whom we serve. So thank you very much. I hope you found some information, certainly as I did, in terms of reading through and researching on genomics. I'm a physician by training, so I'm not a geneticist, but clearly there is much more to come in this space, I'm absolutely sure. And thank you very much.
So I share a question for you. Okay, another question on process genomes.
Okay.
Yeah. From top.
Another question also.
Okay, so one of the questions that's been asked, this is from Sheila Krishnaswamy, is are genetic test kits being overused, used unnecessarily? Some clinics are using kits to manage weight and offering gene testing at a high cost. Is this really necessary unless there's a family history or perhaps an unhealthy lifestyle? So Sheila, I mentioned this as part of my discussion to say that clearly there's value in terms of a personal genome analysis, and that value is varied as we look across various work streams, whether it's how we metabolize medications or drugs, how we react to certain conditions and cancers, or in the space of oncogenomics. On the personal genome space, yes, clearly I think that there's a multifactorial impact of disease that takes place, and into that account, while it's useful to know what's an individual risk in terms of association basis of some of these tests that are coming through, what's really the value, how does it benefit a doctor or a physician in that regard to help him make a more educated judgment. So for example, if we have two individuals who have relatively a normal set of biochemical parameters in place, and one is at a higher risk because of his genetic profile, I think it's useful information for both the physician to know as well as the patient. So there is an extra sense of guard that takes place for that individual to take that into account. Certainly, I think one of the values where we look at the tests to take place certainly shouldn't be in isolation. So as I mentioned, getting a personal genome analysis along with your preventive health check, for example, is a very sensible solution, where you have the physician take you through your basic demographics, your diagnostics, whether it's biochemical parameters or associated radiology parameters, and then link that through with your genome analysis to have a better insight into that process. And that, I think, can be a value add. The other question that came across was from Roshan Prizak, who said, with our limited understanding in terms of non-coding loci and the influence of phenotype, aren't we a distance away in terms of how we look at some of those variants that have come into play in that structure? And really, do we not need more research into that point? And to your point, Roshan, I absolutely agree. I think just as one example that if I may share with you, if you just look at the space of oncology, where I was mentioning that there is clearly some potential in that space. If you look at hypermutated genes, which are identified in the cancer genome atlas, and we identify, let's say, the most common cancers like lung, breast, colorectal, and prostate. We come across almost close to about 300 hypermutated genes that have been identified in that process. But when we really look at which are our driver mutations, which are the ones which are really of intense scrutiny, then we come back to almost coming from those 300, we drop down to almost about 50. And really, in that account, the current targeting on what's currently available in terms of the research that we're doing is barely a handful. So clearly, there's much more to be seen, much more to be read, and much more to be understood within that process. One of the questions that has come from Anup Chakrabarti is how affordable is personalized genomics in a country like India for the middle class? So the way we look at the genome analysis, when it started off, as is invariably been the case when there's new technology or new solutions that arrive, cost certainly can be a hindrance, and it certainly can be a deterrent for a country like ours. The 23andMe test, for example, when it started off at close to about $1,000, and now those costs have come down to almost about $99. Now there are various degrees of personal genome analysis tests that are available in the country, priced in the sub 10,000 rupee category, but there are then other specifics which may be of a higher cost. Again, it's the question of affordability also links into, I think, more in terms of the intended area of use, whether we're looking at more of a proactive use in terms of information that is nice to have, or information that may be necessary in terms of targeting treatment. So for example, in the space of oncology, where we know for a fact that treatment is expensive, there can be clearly a value add which may be more valuable in terms of cost effectiveness certainly in a longer run in that process. So one of the questions that has come about from Nibi Elakat is a pre-test risk assessment and genetic counseling offered to couples before offering them genetic screening, and what kind of tests would be employed in that process? Nibi, I absolutely agree. There clearly needs to be, as I mentioned, counseling that takes place both from a trained professional or a counselor before and after, because there clearly can be an impact in terms of what the results come out as, whether they're suggestive or they're non-suggestive in terms of placing or excluding somebody from a higher risk. And clearly there needs to be a physician oversight that helps, that oversees the entirety of that process. And as I mentioned during the talk, a large number of these include things like when we're looking for thalassemia, hemophilia, muscular dystrophy, a large number of renal disorders. There are a large number of them that take that into account. There's a question that's come in from Amit Gupta as to what's the level at which personalized healthcare is being practiced within the country. I think more and more we are seeing a trend and a movement towards personalization of care. Clearly that's the way of the future in the way we look at how we want individualization or uncoupling of care for that individual basis his needs and basis his requirements to take place. A large number of organizations and healthcare organizations across the country today offer personalization, whether it's in terms of health checks, whether it's terms of their care continuum, disease continuum patterns, whether it's looking at technologies, mobile technologies, devices, care at home. All of those are working towards providing a personalization in that space, in that account. Sakshi Jain is asking, is there any company or any lab that provides testing in that account for cardiovascular disease in India? So as I mentioned within the presentation, Datar Genetics, Sapien are two of the organizations which actually do provide some of that testing in that regard. So one of the questions that has come up is from Mohan CK is do I actually see genomics becoming part of a typical health check? So absolutely. And again, I think what we need to understand is what is the value of a health check that we perceive, whether it's from a high risk isolation or identification perspective to identifying diseases which are in the insidious or asymptomatic stage, and then ensuring there's a regular screening of high-risk individuals, whether it's to do with lifestyle disorders like diabetes or cardiovascular disease, how am I managing my blood pressure, is my weight under control, is my BMI regularly under check, and that there's a follow-up process that takes place. Clearly, when we look at genetic predisposition, this is not an annual affair. This is a once-in-a-lifetime assessment in terms of where one stands on a genetic profiling. But then, as I mentioned, clearly it's important to have all the information available up front through both counseling as well as interface with your physician to provide you clarity and information in terms of what the tests entail, what do they get you, what do they don't. Okay, so Vishal Goswami has asked, is there any age restriction where genomic analysis can be undertaken for an individual? So again, it depends how we are looking at the analysis of the value of the genome analysis. So normally when we're talking about health and a personal genome analysis and a healthy living, we're usually saying it's an adult population. So we're looking at an 18 to 70-year-old. That's the population base that you would be looking at. But certainly the younger age would have much more in terms of interest in terms of understanding how their genomics stand, as well as assessment in terms of the profiling that one has. But if you look at others, where currently there are antenatal testing that happens from a genetic standpoint, which looks at non-invasive measures. So there's within the young age group, we have information, for example, in terms of all the studies that have taken place on cystic fibrosis, and so there is varied value and interface in terms of where you can utilize some of the values of genomics into that space. But again, I come back with the point that it clearly needs to be physician-led and physician-driven. Clearly, if as an individual I was to be told I'm at a higher risk for a condition X, I do need to understand. I need to be clearly informed upfront, as well as after the testing, as to what's the likelihood in terms of the association versus the diagnostic measure of those tests that I'm actually undertaking. Okay, so Mr. Srinivasan has asked that I would like to know if the current information from cancer genomics can be incorporated as part of therapeutic intervention. So as I was mentioning during the course of the talk, absolutely. So we have seen examples of where we've looked at information, whether it's on a impact in terms of the medications that we provide, whether it's looking at chronic myeloid leukemia in terms of medications, or whether we're looking at breast cancer, for example, where there's enough been said about BRCA1 and 2, but that's not the only aspect of what we're looking at. I think we need to understand the entirety of the genome is we're getting to know a little bit about how we're constituted in the process of looking at genetics to drive personalized healthcare. I think there's still quite a bit that we're not aware of and that we will learn in times to come. But I'm sure we said the same thing 20 years ago or 30 years ago when we had limited technology in terms of radiodiagnostics or cancer treatment, for example, in terms of specific radiotherapy or image-guided therapy in that account. I think it's a stepping stone. I think we need to, there is a value, there is a benefit. We clearly need to ensure that our patients are well informed in terms of both the potential benefits as well as what can be the impact on that account for our patients when they undergo those genomic tests. Certainly there's a lot of interesting material out there which I would encourage you to look at and to read. And feel free to connect with me at any stage at all if I can be of any help. Thank you very much and have a good day.
Thanks, Dr. Datta, for presenting this insightful webinar. I'm sure it has benefited to all of us. Uh, Recording of this webinar will be made available on uthealthworld.com. We'll keep you posted. Thank you.